Hello, everybody. Welcome to Town Hall. Good to have you with us on what is a blusterous and gloomy-looking Midwestern winter day. But in my office, it's actually kind of colorful. So at the far end, I got some art. It was a big blank wall, and actually the art is by just happens by one of our panelists today, Joanna Dabrowska. Joanna, can you tell us a little bit of the story of this piece of art?
Sure, I'll be happy to. So this piece has been originally created from a NIH archive photo of Rosalind Franklin working on her microscope. And I actually originally created this piece for one of my PhD student, my first PhD student,
Oh,
Valentina.
wow.
Yes, Valentina Olivera Pasilio, a couple of years ago, and yeah, and I was just inspired by Andy Warhol and by the famous Marilyn Monroe art piece with her. But I just thought that Rosalind Franklin definitely needs another one of those colorful pieces.
So then I found it, been trying to figure out what to do about art in this office. If you haven't, you can see the president's office if you're walking up and down the hallways between the library and the DNA cafe. It's in one of those little courtyards. And it's lovely, although I can't see the sky, so it's pretty gray. It's the gray building on the other side and the gray building inside. And um, so something colorful, because I was thinking about Photo 51, but it's black and white, it's monochrome. And also the images that you can get don't have enough resolution to make them really big. So then in Synapses, I found this piece and I thought it would be, and it is, it's so colorful, it feels good in the office. And our media people printed it on I don't know, some sort of wall sticky thing in two pieces. And our facilities people put it up. So it didn't cost us anything more than we were already spending. Um, and anyway, it actually makes a big difference here in my office. So, so I'm going to get us started. I'm delighted to have uh, Hongkyun Kim, who is a professor in cell biology and anatomy. He has a recent NIH grant on the regulation of calcium voltage gated channels, and we're going to talk about that. And Joanna Dabrowska, who is an associate professor in cellular and molecular biology, and she's part of an NIH grant looking at stress resilience in psychiatric diseases and social recognition. So that's what we're going to talk about today. We're going to talk a little bit about their science and how science happens and works, and it should be a lot of fun. In the meantime, just a little bit of update on what I've been working on and what's been going on. Next week, I go to Washington, D.C. I go mostly because one of our state senators, Julie Morrison, is getting an AMA award, and we're there with some of her folks. She's getting an award really for her support of our Child Advocacy Center. So Archie Chatterjee, our Dean of CMS, put her up for the award. At the same time, I'm going to the Hill. I'll at least be meeting with the staff of our House and Senate representatives for the state of Illinois. So, and spending some time with the American Council on Education. So it should be I'm looking forward to that. I'm looking forward to meeting some of our representatives and our senators or their folks. So I'm doing that. And then we continue our work in preparation for searches. I'm pretty sure that the executive vice president for research is going to be the first out of the gate. We're working on the contract with the search firm, Korn Ferry. The idea being that, sorry, that we get that started in the not too distant future, actually as soon as possible. The vice president for institutional advancement also on the docket. So probably come on the heels of that somewhere along the way. So our work on that both for getting those prepped, getting the search firm, and then also getting the search committees together. Both of those are tightly related to our work trying to sort out the concept of a comprehensive campaign for fundraising for the institution. So I will be talking to the board about whether or not that's a good idea next month. I can't get out ahead of the board, but RFU has not had a comprehensive campaign in the past. They are a lot of work, but they also tend to increase the amount of money that you bring in substantially, and it gives you an opportunity to talk to donors and other folks. So we're, a whole bunch of us are working on what that would look like, how the focus would be, how we would engage the university community, what a big idea sort of conference or collaboration would look like. So we're working and thinking on about that. I'm as a sort of project, not necessarily related to that, but partially, I'm working on RX Kids, which is a cash allowance program for moms and babies. It's been implemented in Michigan. Chicago is working on it. My main concern is that North Chicago, Waukegan, Zion, our friends in Lake County don't get left behind, but it is also been a great opportunity for me to talk to funders and our state level representatives and other kinds of collaborators. So, and also it's a good thing for moms and babies. It increases birth weight. It decreases neonatal intensive care unit stays. It decreases evictions by a lot in the first year of life, and actually even decreases referrals to child protective welfare services. So anyway, I'm doing that. And then finally, the thing that is consuming a lot of work for the university is we're working on the budget. So the board has put forward that we need to give them a break-even budget. Last year was about $1 million short of a break-even budget. It's going to be a bit of work for us to get there, in part because our pharmaceutical costs as an institution went up by about $1 million last year. So not only do we have to figure out how to make up the $1 million, but we have to figure out how to make up another $1 million in pharmaceutical and other health expenses. We are self-insured, so a lot of those costs come back to the institution to pay. I appreciate that a lot of people are doing work. And also that RFU's community has done a lot of work over the years to make sure that our finances are as good as they can be. And I have to say, pretty delighted coming in as a new president with all the work that people have done and what that means and how close we are. But obviously, the more that we withdraw from our endowment, the less money we get from the endowment. It's all the more reason that we have to work on building the endowment and why a capital campaign is so important. But we also have to make sure that we are managing the money that we have. We are tuition dependent, so the dollars we spend mostly come from students. So we have to make sure that we're doing that in a way that helps their education, helps them learn more faster, and helps us have faculty and staff that do a good job for them, which is we have to have a good experience for our faculty and staff. So they can't do a good job for students if we don't do a good job for the faculty and staff. So a lot of people, I want to thank Gavin Ferry and his whole team over in finance for helping lead that work. You will hear more as we work on this over the next month or so. Our main goal for when we get to the board meeting next month is to be able to set tuition. We're trying to keep tuition down. We're trying to, avoid increasing it any more than we have to. So especially we have some programs where clearly our tuition is as high as we can push it. So those are all things that we're trying to balance as we work our way through this. Part of what helps the budget, though, is when people get external grants, like the two scientists that we have today. We not only get to have the fun of doing the work and having that work be a part of faculty life and university life and an opportunity for students, but we do get a little bit of money from the feds as well in indirect costs. I'll just say that overall, universities have to put additional money in. The IDC does not cover all of it, which is a part of what I get to go talk about on the Hill. So across the country, Universities that do NIH work put another 53 cents in to support research for every dollar of NIH they get, including the IDC. It's well worth it. It's a great thing, but our representatives and the government needs to understand that institutions are putting money in addition to what the government spends. That is my update. Questions are always welcome and always more fun. So by all means, feel free to put your questions into the Q&A. We'll get to as many of them as we can. This is my little warning to Hongkyun that I'm coming to him in a moment. So the Q&A, feel free to put questions in about anything, what we're going to talk about next, or anything about the budget or any of the work that I'm doing. But Hongkyun, this is a great project. I remember writing about the calcium channel. Well, I remember learning about calcium channel voltage-gated channels, I guess when I was in college. They are a classic part of how cells work. It's a really important part of sort of understanding the basic biology of our nerve cells and muscle cells, heart cell, like a whole range. Everybody uses this in the body. But can you tell us a little bit about your work and the grant?
Okay, so today I want to share with you a project which is really basic science of the neuroscience, but surprisingly has some clinical implications. So we are studying the CAP2 calcium channels in C. elegans. C. elegans is a genetic model organism. So at the end of this presentation, I hope you are convinced that why research on CAP2 channels in C. elegans is actually very helpful to some of the very challenging neurological disease. So let me start with some background. So neurons communicate at the tiny junction called the synapse. And when the electric signal travels down to the terminal end of the neurons, then they need to actually propagate this signal to the next neuron through this tiny gap. Okay, so if you look at the terminal end of these channels, then you're going to see tiny vesicles contains chemical called neurotransmitters. And so this is where calcium channel, CAP2 channels comes in. So CAP2 channels sit at the tip of the terminal and then play as a gatekeepers. So when electric signal arrives, then the CAP channels are activated. And that activation causes actually the opening of the channels, and the calcium comes in, and that calcium mediates fusion of this neurotransmitter containing vesicles. And that release the neurotransmitters, the new cycle of the synaptic transmission. That's actually how they work. So without this CAP2 channels, you can see if that's not working, then neurons cannot really communicate, right? And also, if you have too little activity of the channels, then you cannot really propagate signals. Conversely, if you have like too much activity, then the circuit will be overloaded. So getting the level of these channels just right is the critical for the normal brain function. So why does this matter clinically? It turns out that if CAP2 channels are dysregulated, then consequence can be very severe. Too little or too much CAP2 channel both link to the like ataxia or severe epilepsy. So both end actually, right? So this is what we call Goldilocks problems. So we need CAP2 channels to be just right. So here's the actually critical gap in our knowledge. So even though we know actually this dysregulated CAP2 channel functions or levels can cause of the many types of actually this neurological disease, but we don't know actually how these channel levels are regulated. We really have no idea. So then you're going to ask actually, why are you studying, CAP2 channels in C. elegans? And the reason is that genetic complexity in mammals. So as you can see here, the CAP2 channels consist actually all the calcium channels, all the voltage-gated calcium channels, including CAP channels, CAP2 channels has actually three proteins actually, and in mammals actually they have multiple copies. So instead of actually single copies actually, you have 48 different combinations. So that makes really almost impossible to study.
Mammals have 48 and C. elegans has 1?
Only one, yes, right. So
So
C.
just for C. elegans is a roundworm, right? It's not so round
Yes.
really necessarily, but it's a little worm. It's really, really well understood. I mean, people have been studying C. elegans forever, and all of its genetics is known. All of its cells are known. Okay. Anyway.
Yes.
Yep.
Right. So yeah, I mean, people just don't realize, but actually, 8 people got actually Nobel Prize for studying C. elegans. So that's like, so well studied. So yeah, so basically in C. elegans has only one, kind of actually channel. So that makes really easy to study. But additionally, the C. elegans has only 3 days of life cycle. So you put actually single worms on a plate and you come back 3 days later, you find actually 200 animals there. So, it's very easy to study. So also then makes actually you can do large scale genetic screens. So, you work with mice, then it's not really imaginable. But also, a lot of, because it's studied so heavily, a lot of genetic tools are developed, and you can, to actually make all kind of actually genetic changes. So that makes actually very good model, particularly. But yeah, the genetic simplicity is the most critical for CAP2 channel studies in this case. So then let me walk through actually our approach and what we expect to learn from this. So our strategy has actually 3 steps. So first, we generate the animals that fluorescence tagged actually the CAP2 channels in worms. So we can literally see, how many actually channels are there. So we can use that. And then actually second, we actually do unbiased mutagenesis screen. So basically we randomly mutate actually all the genomes and find the worms has actually some worms actually higher level of calcium channel. Some animals has lower level of calcium channel. So we find those kind of mutant. And this mutant worms will provide actually, how these channel levels are regulated. So, so we can find that. And then once we find this kind of mutant, then we actually sequence entire genomes. And then actually using bioinformatics, what gene is responsible for this kind of, variation in the levels. So that's how we can, study actually. So in terms of scientific impact, we will reveal how these channels are going into the synapse and also how they degraded, molecular levels. So that's actually what we're going to find in scientific but also translational potential in terms of translational potential. So genes we discovered becomes candidate therapeutic targets. So for instance, that some of the actually genetic variant cause gain of function. So it means that calcium channels are overly active. Then we can actually use actually some of the genes that when genes are inhibited, then reduce the calcium channel, then we can actually develop some kind of drug targeting that gene. Then we can see a reduction of calcium channel levels. So that will actually ameliorate the gain of function phenotype. So conversely, some of the genetic variants cause actually reduction or loss of function phenotype of CAF2 channels. Then we can actually find a new drug that, the stabilizing actually this calcium channel. So then we can actually apply those ideas. So that's actually what we are trying to achieve. So the bottom line is that basically we are using, forward genetic screen in C. elegans, and then find actually the potential therapeutic target for calcium channel, disease. Okay, so that's actually generally what we are doing.
So what can you describe like an experiment that you imagine you guys will be doing, just in general terms?
Oh, so yeah, generally, yeah. So I mean, we actually have this already actually CAP2 channel tagged actually animals already in hand. So we also have some patient mutations. For instance, that there's a familiar hemiplegic,
Okay.
hemiplegic migraine disease. So there's
Okay.
actually a mutation for these calcium channels. So we also introduced those mutations in this worm. And so it's a gain of function mutation. So that means actually channels overly active. And so does C. elegans actually. So worms also shows that, channels are hyperactive. Even though we don't know actually they shows migraine. But but the
The
but the
roundworms don't complain of migraine. Is that what you're telling me?
yeah, but the idea is that then we can actually reduce calcium channel levels. That way, we can actually suppress actually this kind of disease symptoms, right? So yeah, we find some mutations that help actually reduce channel levels,
Right.
and then may actually potentially you know, possibly actually has some therapeutic implication actually.
So I think you've actually just answered this, but Hector asks, how do you talk about or justify using C. elegans as a model for a human relevant disease? Obviously, humans are billions of cells. C. elegans has, a couple thousand cells and 300 neurons. So how do you move from this is what we find in this model organism that is so much easier to work in than people to a target that, might be interesting in human disease or human conditions?
Yeah, so well, yeah, kind of seems obvious, but because actually some of the biological process are not too unique to human,
Yeah.
it's very conserved. So like it's this kind of actually channels. They are really conserved function, and so uh, in actually these channels also is critical for synaptic transmission. So does mammals as well. So it's very conserved actually process, and so does actually like the proteins regulating. These proteins are very conserved. So once we find actually target, then we can apply for actually mammals, including humans. So that's the basic idea. But because it's a simple and also it comes with the robust actually genetic tools, that's actually our justification actually.
Yeah, I mean, I think there is some reason that people have been trying to push science to be, in humans, but we're too complicated. And
That's right.
we don't reproduce every 3 days, and you can't take all of our neurons out. It's just all the confounding variables make it so very difficult to understand at a basic level what's happening with any particular protein or anything else that That's why models like C. elegans are so important, just to understand how these basic building blocks of neurobiology work.
Right. So yeah, there are actually, very critical, clinically important problems, but with actually some of the models, including humans, we cannot really answer those kind of questions. So that's why actually some, model systems are very good for actually this kind of actually, approaches.
So Emil Rosenkrantz says, "Hey Hoki, I love hearing about your work. Thank you for sharing. Is there a way in C. elegans to limit mutation to a population of cells, maybe differentiate deficits in the worm brain versus the synapses to the muscles?"
Oh, yes. I mean, yeah, basically, of course, we could do that. Actually, there are tools that, so what I showed here actually showing entire actually brain, but we couldn't limit actually very specific types of neurons. And yeah, because actually in C. elegans, cell lineage is really literally from the fertilized egg to the entire animals are mapped.
Yep.
So basically we can, modify actually those things. Yeah, we can actually, yeah. Currently we are working with the some other models. We are actually limiting those actually mutations only in a certain type of cells. So yeah, those are actually commonly done in C. elegans actually.
Yeah, I mean, I think actually at the blastocyst stage, right, which cell is going where, and you can
Yes, right.
swap cells in and out. And not that you necessarily would do that for this, but people have done that, as I recall. Michael Rosen asks, Hoki, can you discuss further how this may help the development of new drugs to combat some of the diseases you mentioned? As I recall, gabapentin works on calcium voltage channels, doesn't it? Or maybe I should ask the PharmD that question.
Yes, I believe so. Actually, that's working to the outside of this. There's actually, I mentioned the three subunit, and there's actually extracellular subunit. That's where actually gamma pentin works. So that, yeah, that is actually well known.
All right. Joanna, you wanna talk a little bit about your work, and we'll come back and and maybe even Hongkyun can has if you guys actually have at some level, I'm betting, overlapping sets of interest given that you're both doing neurobiology here.
Yeah, and I think it's even more than that, just talking about the gabapentin, which is obviously not just is not just the drug used for like neuropathies, but it's actually has also been approved. And it hasn't been approved, but it's used off-label for
Yeah,
anxiety disorders as well.
yeah, and maybe mood stabilizing, even though I don't know that it really does anything.
Yeah, yeah. So I think there's definitely more common than we think. Let me try to share my screen with you. Can you see everything all right?
We need you to go to the slideshow.
How about now?
Yes, perfect.
Okay, thanks so much. So yes, thank you, Hoki, for sharing your work. And I also cannot emphasize how important the fundamental basic science research on calcium channels is and how much we can actually learn from that. What I'm going to talk briefly is the research project that we have just recently got funded for the role of oxytocin, which is this hypothalamic peptide and hormone. And this specific project is looking at the modulation of the supramammillary nucleus. So that's a part of the hypothalamus, like the posterior part of the hypothalamic brain region. And that's an NIH project. That's a 5-year project that has been actually spearheaded by my collaborator from Mount Sinai School of Medicine in New York City, who is an expert in a social recognition memory. So, just something, in terms of the fundamentals of this research question is that we know that social recognition memory It's just absolutely fundamental to our ability to acquire, retain, and recall identities of our fellow members and peers and family members. So this is fundamental in a way that the social structure depends on it. Our daily interactions depend on it. And it's not something we typically often think about during the daily interactions. And something that is very useful for us from a science perspective is that this is a very conserved type of response across species, including mice, rats, and of course, essential for humans. And from a human health perspective, and from the NIH perspective in particular, the important thing is that it is very relevant to many of the disorders, neuropsychiatric disorders like schizophrenia, Alzheimer's disease, and autism spectrum disorder, and other disorders, neuropsychiatric disorders as well, because we know that those disorders often present with a deficit in the social recognition, not just in the social interactions, but specifically in the social recognition, memory, formation, and recall. And generally speaking, we know quite a lot about those mechanisms, but many of the cellular mechanisms, specific circuitry that are really underlying this form of memory are not very well understood. So we got, between me and my collaborator, we do share this profound love for one of those peptidergic hormones and neuromodulators, oxytocin. So I'm sure you've heard about this peptide before. It's produced in the paraventricular nucleus of the hypothalamus, in addition to other hypothalamic nuclei. It's known profoundly for its effects on the peripheral physiology regulation. So the uterine contractions during labor depend on oxytocin release and oxytocin receptor, milk ejection reflex. So this is a typical hormonal role of oxytocin. However, we know from decades of studies now that oxytocin release from these hypothalamic nuclei also to the central nervous system, including the limbic system, has been really fundamental for many of the complex behaviors. Oftentimes, those behaviors are related to affiliative behaviors. So they can be maternal behaviors, alloparental behaviors, pair bonding formation, partner recognition, a lot of social recognition related behaviors also related to parenthood in general. So we were really interested in this peptide specifically in its relation to social recognition memory. So when we first started working on this supramammillary nucleus of the hypothalamus, we basically found that there is a very robust expression of this oxytocin receptors in the SUM, for short for the supramammillary nucleus. And what my collaborator was able to show, and she actually published it last year, she was able to show that if you block oxytocin receptor transmission in the SUM, you actually completely abolish the social recognition memory. So the animals, the rats in our case, they no longer recognize their peers, and they all treat them as novel rats. So based on this, and based also on the fact that some of the previous published studies in mice show that the SUM projections to the CA2 region of the hippocampus is also necessary for the social recognition memory. In fact, inhibition, so the decreased activity of this pathway has been shown necessary for proper social recognition. So based on all of this information together, we come up with this hypothesis, or this circuit hypothesis, in which during the social interaction, there is a release, which has been shown, of oxytocin in multiple brain regions, including in the supramammillary nucleus. Once oxytocin is released, there is an increased activity of the local GABAergic interneurons in the SUM. And then what these interneurons, these GABAergic inhibitory interneurons do, they basically inhibit the projecting neurons, the projection neurons from the SUM to the CA2. Now, because it has been also shown that those projection neurons target local interneurons in the CA2, what it basically means that now these interneurons are disinhibited. So now their activity is reduced. It's actually reduced. So they're inhibited. Their activity is reduced. And then the pyramidal neurons, finally, which are responsible for the social recognition, memory formation, now their activity is increased. So this is a model that we propose, and this is the model that we are testing currently. So we use a variety of different techniques. So we use very basic techniques like in vitro patch-clamp electrophysiology, slice electrophysiology, when we can manipulate oxytocin receptors while recording single neurons activity. And we can basically learn a lot about how these neurons respond to oxytocin application and oxytocin receptor activation. And we can also figure out a lot about these neurons, not just cellular responses, but also their synaptic connectivity. So really, we are trying to understand very basic cellular and synaptic mechanism that would underlie this effect on the SUM level. And then we are—
Hang on just a second. So for people who don't know what a patch clamp is, it is essentially the world's smallest pipette that then gets attached to a cell, and there's a little wire in the middle of it, and the little end of the pipette isolates those cells on the membrane, and the little wire lets you get sort of an EEG, except it's not in of what's happening to that cell as it gets stimulated or not stimulated, right? So that's how you guys are measuring the activity in the neuron, whether you've changed this after you've manipulated or before you've manipulated the receptor. Is that fair?
That's very fair. And
Okay.
we can do that with addition of pharmacological manipulation. So
Right.
something before and after the treatment, and then see how excitability, how differently the neurons are responding, how much they spike, how much more they're spiking. We can also do that with addition of some newer techniques like optogenetics, in which we can release or like simulate the release of the neuropeptide coming from the terminals. And then while patch clamping, see how the neurons would be responding. So there is variety, but that's a very fair explanation of the actual, patch clamp recordings from these individual neurons in the SUM. And something—
Yeah, I just want to point out how kind of astonishing this is. You take a little piece of glass that's hollow with a little wire in it, and you stick it on a part of a cell, right? And you actually try to control what part of the cell you're on. Like the level of technique and technical parts, and patch clamping is old. It's been around for decades. But anyway, it just always amazes me. And then optogenetics, is that when you put in a little phosphor or something like that genetically into a protein, and it lights up when it's active or not, and then you can watch what's lighting up? Is that what's going on?
Yes, exactly. So for example, in this particular case, we would express these light-sensitive channels like channel rhodopsin in, for example, oxytocin neurons in the hypothalamus, and then these neurons would project. So the channel rhodopsin would also be expressed in the terminals in the SUM. So then when we shine the light on the SUM with the specific protocol designed to release the peptides, we can then release oxytocin and study not just application of the artificial oxytocin peptide, which of course is very important, but we can also test it on a kind of more physiological endogenous terminal release level. So that's exactly the idea behind those studies. And then of course this is extra. I mean, this is in vitro. This is slice electrophysiology. It's like you said, it's a fundamental, relatively old technique which gets us to the single cell resolution, and and we can study the intrinsic circuitry. Whereas we really wanted to know what's happening in vivo, and that is something that is in
the animal,
in the animal. So in this case, we also are adding the techniques like, for example, fiber photometry recordings, in which you can then record from the very specific population of neuron via the optic fiber implanted in the via the cannula directly in the brain region of interest, while the animal is performing behavioral tasks. So in this particular case, the animal is performing the task of social recognition test, which is this little diagram on the right. So basically the animal is able to recognize between the familiar and novel peer, based on a very simple idea that the mouse, the rat, would basically spend always more time with the novel rat, will spend always less time with the familiar rat. And when we see lack of this discrimination, it's a very simple proxy for deficits in social recognition deficit, of social recognition memory deficit. So this is one way of trying to record the activity of neurons while also measuring the behavioral responses while they are performing the task. And then to kind of flip this, we can also introduce some of those techniques like the one called chemogenetics, in which we use designer receptors on a specific population of neurons, and we can manipulate them artificially by introducing specific ligands to these artificial receptors. But this would allow us to drive the activity or inhibit the activity of the specific population of neurons, and then see how this affects the social recognition memory in these animals. So kind of, using the causal versus the necessity of some of these neuronal populations, and how this can be manipulated.
So my family had pet rats, and we're great fans of rats. You said that rats are more likely to spend time with the novel rat or with the familial rat?
That is an interesting thing. They actually prefer, so they are driven by novelty. So they will always try to explore and investigate.
They are curious.
The novel rat more. And that's why by seeing a rat that doesn't really discriminate or maybe spends more time with a familiar, like treating him as it was a novel rat again, it tells us that there might be deficits in their
ability to recognize social. Okay, so I'm just kind of curious on the scale. If you have, I don't know, 100 rats, how many of them actually spend more time with the novel rat than the familial rat? Is it a big difference? Is it like 9-to-1, or is it relatively subtle, 7-to-3 or something like that?
I can't tell you the exact numbers from the top of my head, but this is a relatively well-established, and it's used in both mice and rats. So I think that the response is fairly robust. So this preference for the social novelty is a pretty robust phenomenon.
So, there's a lot of, I don't know, silliness in the world about, especially among, I'm just going to say it, among physicists, that, like, why haven't biologists figured out more? And this is a part of biology is just so much harder than physics. So I spent time in a chemistry lab, and we're working on the order of Avogadro's number, 6 × 10⁻23 molecules in an experiment, right? And so if you're doing an experiment on this in rats and say 90% of the rats are supposed to behave one way, not only do you have to get all the technical stuff right and have a robust biological finding, then you have to be able to see it by repeating it enough times in enough rats that you have some statistical scale to your work. It's remarkably difficult to get answers in these biological systems.
It is remarkably difficult, and I also think that something that we have to remember about is that because many of those circuitries and many of those cellular responses can be common for several or multiple behaviors, right? that we have to have a way of really distinguishing between one type of the behavioral output versus the other type. So for example, in this particular case, we know that SUM projects, just to mention the two, it projects not just the CA2 part of the hippocampus, it also projects to the dendrite, which has been more involved and has been more implicated in the regulation of the spatial memory, for example.
Oh,
So really to be able to distinguish these two pathways, we have to be very careful. So something that will be the first thing to do will be to even determine, okay, is this the same population of SUM neurons that project to the CA2 and the dendrite via axon collaterals, perhaps, or is it this completely independent population of neurons? So a lot of questions and a lot of control experiments have to be really added to figure that out.
so actually, Hector asks almost a confounding variable question here. So Hector Rosgato asks, can you rule out the possibility that blockage of oxytocin receptors affect olfactory function? And rats are driven a lot by what they smell. That may be actually how they're recognizing, for all I know, familial and non-familial. But like, how do you manage, or do the different senses play a real part in how this biology works?
Yeah, that's a great question, because it's not just obviously just the visual for
Yeah.
for rats. I mean, they look all the same to me. I'm sure that they can see more than we do, but you're right. I mean, the olfaction is the number one stimulus for them. I think that there are ways to rule that out. So I remember we actually talked about it. One of our speakers a couple of years ago, Jane D. Bynes, who does a lot of those type of interactions, and he even done some, temporary anesthesia with lidocaine.
Oh yeah.
Then in the nose of a rat
Yeah.
or mouse to really rule out this contribution of the olfaction. Or maybe it is an olfaction, but
Yeah.
it doesn't still mean that because it might be a part of it, right? It might be part
Yeah.
of this whole process. So
It could be the most important part. Like it doesn't have to. So, and maybe Elliot has a similar kind of question. Are there sex differences in oxytocin CA2 circuits that contribute to social recognition?
yeah, unfortunately, we don't know that yet. We are literally starting to record. We started with recordings of the basic properties and excitability of these SUM neurons without even looking at the CA2 projections yet, simply because there is not a single study in the literature on the properties of those SUM neurons. Not to mention, there is absolutely nothing about the sex differences. So this is definitely on our radar, and this is definitely something that we are already recording from both male and female rats.
Great.
My very final slide is just a big thank you for all of the lab members that had contributed to producing, a lot of preliminary data for the grant submission, or I should say, 4 grant submissions to be clear for this particular funded project. And I also obviously wanted to thank my collaborator from Mount Sinai, Hala, Haron, and Nicholas, who was who approached me to really get this project rolling. So thank you very much.
Thank you. And of course, as always, if there are more questions, if people have questions, feel free to put them in. So, Hongkyun, I'm thinking about C. elegans and the difference between the brain and the periphery and how those synapses work. That is if you think about Joanna's work, right? You're talking about the little terminal parts of each one of those neurons and the synapse across as they're talking to each other. How do you guys, as a field or as biologists, how do you decide, I think I'm going to focus on this particular piece at the end of the synapse, as opposed to I'm interested in, or I'm going to be looking at how these larger systems work? what part of your career or what makes you, why do you think in retrospect that you ended up in these two different parts of biology? I'll ask each of you that question.
Okay. Yeah. So, well, the reason is that actually, because while we are studying, I end up actually working with the, one of the actually earlier study leads to the calcium channel. So that's why actually we
That's
started
where your
calcium.
fascination with it came from.
Right,
Yeah.
right. And then actually, in reality, I'm not just interested in calcium channel, but surrounding area as well.
Yeah.
And so that means actually synapse itself, how they are organized, how they actually changes depending on activities. So that's actually how it leads to. But yeah, also we can study, I mean, in the field actually, just because C. elegans is small, means that actually you have to study synapse. So some people studies actually more like behaviors. So Joanna mentioned about the optogenetics. So in some people actually, in at least in one field, so we can actually regulate their movement actually using optogenetics. So make them go forward, reverse, that kind of movement. So some people actually studying, because also is neurons are a small number. So we can actually study their networks. So some people, dedicate their research on that field. But in my case, I happen to be, I found actually these channels. So I'm interested in that area. So I'm looking at, more like that areas. So that's how I end up.
Great. Joanna, how would you answer that question?
So honestly, my general interest has always been in mental health and neuropharmacology, neuropsychopharmacology. I would say that, specifically to peptides, how did I get into this field? I had a strong history of peptides researchers at Emory where I was doing my postdoc. Were a lot of people working on maternal behaviors, social behaviors, and I had very little interest in that at the time. I actually had seen several presentations from one of my former co-mentors, Larry Young, and he was fascinated by oxytocin in a maternal behavior, showing how a single injection of oxytocin into the brain of a naive virgin rat, female rat, can induce
Maternal behavior.
spontaneous alloparental behaviors within matter of, several, maybe 20 minutes. And I just remember that, and of course, we know that naive females don't display those type of behaviors. Virgin females don't display those unless they are kind of primed with the whole with the mom and the offsprings. But I just remember that this just blew my mind, and it kind of like took me to this place when I realized that the peptides as in neuromodulators and the way that we understood them, those are powerful modulators of behaviors as well. And I think this is something that kind of brought me to this field.
So Michael Rosen asks each of you, how do you think your research, with the pathway from your research to patients, what would that look like? What kind of stuff has to happen? What do you have to find? What other steps do people need to do?
I can start maybe because oxytocin has been actually excessively tested as a potential pharmacotherapeutic.
And it is, Pitocin is a drug that we give people.
Yes. So Pitocin for the gynecology obstetrics, but also oxytocin and the derivatives as an intranasal spray.
Yep.
They have been tested for, not just psychiatric disorders like PTSD, and they've been also tested for the autism spectrum disorder as a kind of a booster for the social, interactions. So I think that, this is already happening. This is already happening in many disease models and many different neuropsychiatric disorders.
Okay. Yeah. So yeah, in my case, actually, my research is more fundamental issues, but yeah, I think there is actually the opportunity for already we find some genes that might be relevant to humans, but the only issue is that we have to really verify and, with actually human neurons, right? So that's actually also, we are due to the list, but yeah, so then once we identify, can we see actually in human neurons, whether actually this can be changed, then actually we have to go through the, some kind of drug screen. So long way to go, I would say. Yeah.
Well, yeah, but like as you said, I mean these channels don't exist on their own, right? So
Right.
it's not like the membrane just has this channel in it.
Right.
It's essentially a soup of
Right.
all kinds of proteins, some of which are moderating that, some of which are may have little or nothing to do with it, but these a voltage has to get there somehow, right? And that voltage comes usually from sodium channels, but like there are all kinds of things that play into how these work. And as you said, you're interested in the synapse as a whole, not just on this particular channel. That I mean, you never know what you'll find in amongst all of that stuff, or yeah. Well, so let me ask you each something you're excited about out of this work. What do you have the money now? You have to do the work. So what's coming up in the work that excites you?
For me, it's a completely new brain region, something that I have not been studied before. I would have probably not studied it unless my collaborator at the time approached me and just convinced me to focus on this brain region.
Give it a whirl. Yeah.
It's a completely uncharted territory for us. The exciting part is that whatever we discover is going to be a new and novel finding. So, in this sense, at least especially on the synaptic and the cellular side, this is definitely an exciting era.
Yeah, so our approach is not like, targeted really. So as I mentioned that, the for the genetic screen is that it's like chocolate. You don't know what you're going to get actually, right? So that's actually how it's approached. So, working with actually new, unactually imagined,
Mhm.
those aspects. So we're going to work with actually something new. So that's actually what really excited me.
And then Susanna Calkins says, I don't really have a question, but I appreciate hearing more about the research and also had lab rats as pets when I was a kid. They're great. Like if you get a chance to have a rat as a pet, you just have to get used to the tail. That's all. So I want to thank Hongkyun Kim who's a professor in cell biology and anatomy, and Joanna Dabrowska, who is an associate professor in cellular molecular pharmacology. Thank you guys for being with me today and talking about your work, and thanks for all the great work that you've done to get to this point.
Thank you very much for the invitation.
Yeah, and thanks to all of you for joining us today. Thank you for all you do to support our faculty, our staff, and our students. It is the work of people like you that make the university such a special place to be. It is Friday. I hope you get to have a weekend. Take care, everybody. Bye-bye.